Welcome back to Imagine More, Create Solutions. I'm your host, IMCS co-founder and Chief Scientific Officer, Andrew Lee. In our last episode, you'll recall our chat with Dr. Scott Kephart, President of Quasar Instruments, where we talked about the importance of investing in high-quality reagents and stress testing your validations. In this episode, we dive into the impact of poor quality and false lab results, especially for smaller and physician-owned labs. Let's jump right in. Again, we've talked about price versus value, and we're kind of hounding on value, not only on the differences of the enzyme itself, but paying attention to the details. How are our vendors' pricing structures? Is it per ml or per sample? But again, I think that's only on the consumable side. If we kind of pull that back again to what sort of value is that bringing to the table, and why should these operators actually pay attention to the value cost rather than just the price of the consumables itself?
When we run our financial engine, one of the things we have to explain to them is the finances of a lab is 85% of fixed cost business. What that means is that when you run a variable cost business, when your productivity goes to zero, a fully variable cost business, when your productivity goes to zero, your costs go to zero. And when your productivity is high, your costs are high. So it ramps. In a fixed cost business, those costs are fixed, whether you're running samples or not running samples. And when we look at a detailed analysis of the profitability of the business modeling, cost per samples are difficult to calculate in LCMS. They're probably running around $10, $15 per sample in consumables, depending on how you want to do that. Income levels per sample run are probably running. It's highly dependent on the payer mix of the customer, their testing policies. It's a very complex analysis, but let's call it $100 per sample. So when you look at the business aspect, if your cost per sample is $10 per sample and you save 10%, which is a huge savings, you're saving $1 per sample. So your cost went down from $10 to $9, which is a big percentage change, and any purchasing agent would be excited about a 10% savings. Your income went from 90 to 91. It didn't really change the income that much, and you're introducing variables that might affect that whole $100 income stream. And that's where, when we model, our engine will model downtime, it will model the cost of downtime, it'll model kind of the true cost of this business, and that's really critical for them to understand that really saving $1 can end up costing you $100.
The cost of reruns, the cost of retests, and the cost of false negatives.
It also sounded like you have to pay the payers reimburse the payers if you've reported out false results. That's going to be huge.
Many labs have been shut down because of the fines associated with putting out bad results by Medicare. You not only have to pay it back to Medicare, they fine you. And then there's also the lawsuits that the payers produce, because you have to return their money and they want interest on it.
Yeah, it's, and the difference, you had mentioned reruns, and you had mentioned, reporting negative results. A high quality lab with a high quality oversight will catch their own errors. That's what triggers a rerun. If you don't have the quality in the back end to catch your errors, robust quality controls, those type of things where you can detect errors, then you're actually releasing false results, and that's when it gets scary fast. It's interesting. I usually I have a technique with our financial engine of battling I could get this cheaper, I could save 2 cents there. Actually run our analysis at cost per sample of 0. So I just say, okay, let's forget saving money. Let's say you got all your reagents for free. You didn't pay for a single thing. Look at the impact that makes. And they know that's ridiculous. I do it on the cost of instruments too. But if you compare that to the cost of not running, you can't save yourself the profitability in this business. You have to keep running, and you have to produce quality results, and you have to have customers. The commercial labs, if you produce bad results, you lose customers. And those customers are extremely valuable and hard to replace.
Well, the thing is, the more tests you run, the
More
larger the
frequent.
mistake is.
More frequent dosing. Yep. We covered a lot on the enzymes as well as the cost of quality, but then are there any other aspects? Say, for instance, buffer. Would that be also something to be paying attention to? I mean, the devil's in the details, right? Claire, anything about buffers?
Enzymes work within a specific pH. Urine samples range between a pH of 5 and 9. And most enzymes work in a very specific small range, say 5.4 to 5.6. Each enzyme is different. So if you don't have the correct buffering capability, it's not made correctly, your enzyme's not going to work efficiently. And you're not going to hydrolyze your samples. Also, in the mass spec, if you do sample cleanup, you need the correct pH. If your pH is wrong, you won't clean up your samples, you won't collect the analyte of interest at all. pH is really important in LC-MS technology and sample prep of urine.
The other thing is, you mentioned inhibitors. We published a paper on that in JAT a few years back. The pH of urine is already 5 to 9, and the pH of the buffer is important. And the enzyme performs at a very specific pH range. Your extractions or your analysis on LCMS all perform at a very pH specific range. But we actually showed that you need a certain large quantity of buffer to start to dilute out, simple compounds like urea. Urea is a common denaturant, a protein denaturant that's in urine, right? That chemical was just discovered in urine, and that's why it's called urea. There's a lot of it in urine, and it is classic for protein denaturation. But if you don't dilute enough of it, then you might be just messing up and denaturing or unfolding or destroying your enzyme that you paid so much for. So these processes and these robust testing, these are things that we've done at IMCS, not only on the enzyme side, on the pH side, as well as on the buffers and the dilution of the urine samples to make sure, Scott, he has one less thing to worry about, right?
And that's a good point, Andrew. We have many independently published papers that stress enzyme testing, adding lots of glucuronides to each sample and incubating them with our enzyme and then testing. That's our point in producing this data, this information for customers, is stress testing our enzyme, and we have the data to prove it. Anyone going on our website can read. Independent laboratories have tested our enzyme, and we've done our own internal testing too. And it's also great that Scott has tested our enzyme and stressed it and doesn't have to worry about it.
There's another component to it, like you mentioned, these little details. We did cover that maxim or that adage about the devil's in the details. There's a lot of technical details on this complex analytical workflow. Some of the things I can bat off right off the top of my head is like a buffer's choice. We were just on buffers, but Claire also mentioned salts. Buffers are salts. They are salts to maintain a specific pH. But if they are incompatible with the mass spec, you're adding a lot of buffer or salt and try to control your pH, which you have to do, but then you're also mucking up your systems because by adding those buffer salts. But, again, for our in-house R&D and our new product with the RT, we've actually used a volatile salt. Volatile salt meaning that it vaporizes upon ionization with the mass spec. So acetate is a volatile salt, vinegar, right? And you can smell it and it kind of vaporizes. So it doesn't actually crumb up your system. That's a fantastic buffer to use, acetate buffer. But we've seen in the market some of these tris or EDTA salts that are used in there, and those are really, really non-volatile, or they will muck up your mass spec. Those are salts that you should not be using.
So Andrew,
yeah.
tris and EDTA will actually clog your system, clog your columns, or clog your source. So you have to clean that constantly.
It's probably a multitude of different problems. I won't pinpoint to exactly what, clogging systems or any of that, but then tris is a very strong, it's one of the goods buffer salts. So it will not vaporize on mass spec. Or it could actually form a counter ion. So like you could have a sodium or a tris salt instead of just a proton. So instead of having that analyte with that specific mass that you're analyzing, it could actually have a completely different mass and you no longer see it on your system. And that could result in your false negatives and so forth. But if your LCMS is running well and it's already been stress tested, all those should be addressed. And, again, like you said, the doctors should not be worrying about this. Oh crap, is this a volatile salt? Is it not? Well, from IMCS with Scott, it's already a volatile salt. You don't have to worry about it. This is the system that we've got you prepared on. And you've kind of covered on a interesting side of things, right? Those details that they don't have to worry about. It's sample prep, right? I'm sure you guys get a lot of calls on that. I mean, do you want to kind of touch on that?
Yeah. It's funny. We've been talking about stress testing and stress, and it's hearing this talk about it, I mean, I just realized that we do controlled stress testing to remove stress later. The stress of the instrument being down, the stress of having results, the weird ionization that you talked about, or the unexpected ionization that you talked about when you start failing QCs and you start failing this and you don't know why, and you have to do the stress of figuring out what's going wrong, either bringing an instrument back up, calling a repair, cleaning it, venting it. All these things stress out, especially our customer base, a lot. We're kind of a frontline defense for our customers, so they call us for service calls, and then they'll call an LCMS service center. But always the mass spec is broke. When we call the mass spec broke, and 90% of the time it's a prep error. And I would classify, poor reagents into a prep error kind of classification. It doesn't have to necessarily be human error. But yeah, most of the time, most of these issues that we deal with can be traced back to some sort of error or something happening during the prep process.
Well, we covered a lot. I mean, the right process, the right procedures. I like that, that line, controlled stress testing, so that we can reduce future stress. I think we should do a lot of that, actually. We try to avoid it, the pain up front, thinking that we can just kick the can and it will come back to bite us.
Yeah.
So Scott, based on today's discussions, are there any key elements that you'd like to summarize and leave for the practices for our audience?
Yeah, and I'll focus on the POLs, the smaller labs, and the new labs. They want to start out with a robust process. That process includes methodology, instrumentation, analytical procedures that they're using, and the consumables that they're buying. Actually, a lot of people think that when we sell this, their paradigm is the FDA approved instruments, and so they think the validation is going to take a week, and it's just checking something, and they don't understand that this is an LDT. Unfortunately, the analyzer world, the FDA, they use the word validation. We use the word validation. What we're doing is really what they had done for the FDA combined with an IQ/OQ that they're doing on site. So it's very, very challenging. Getting the lab started with the right processes, the right consumables is critical. The first 6 to 8 months of the lab operation is absolutely critical. It's during that point that confidence will be lost. A new reference lab, a commercial lab, they have new customers. So if they mess up in that 6 to 8 month period, they're going to lose those customers that they invested heavily in. A lot of my customers, because it's a POL, because it's an in-house lab, they don't think they have to worry about customer care. They absolutely do. They have independent physicians. They have physicians working with them with opinions. And if the physician staff doesn't have faith in the results they're getting from the in-house lab, they're going to be very, very vocal about it, as they should be. They need quality results. Going back to the lab, if you have the right processes, it's so much easier to start out with the correct processes, to breeze through your first audits, to establish policies and procedures that give you the quality consistency that you need. It's very difficult to go back and fix a lab that is having issues. Those are very challenging. Kind of the last point that I'd like to make, and it was brought up in this talk quite a bit, but I really, really try to iterate it. I iterate with my staff, our customers. We always talk samples. We always talk urine. We always talk the mass spec samples. At the end of the day, these are all patients. These are all patients that decisions are going to be made for them. We do a lot of customers in rehab and pain management. That's the main markets we focus at. I'm terrified of being associated with a lab that releases false results, false negative, false positive, to those patients. In the rehab world, you have somebody that's going through a difficult point in their life. They're asking for help. The thought of falsely accusing them of a false positive of something that they didn't do at a low point in their life terrifies me. So that's kind of, where we focused. I glanced over that at the beginning, but being patient-centric, really developing policies and procedures focused around the patients is critical, not only to the patients, it's also critical to the long-term success of any lab. If you try to make decisions that are not patient-centric, in my experience, you get in trouble. It's not a question of if, it's a question of when.
Oh, that's a completely different perspective. We all live in our own world, sort of a bubble. I mean, I myself would be more of a scientist looking for the latest and greatest enzymes, what sort of features or characteristics that have. But if you trickle further into it, it is really patient-centric. We need to do the right thing, right? It's not just the cost per sample. It is that value that we need to start looking at. Embracing, the client's perspectives, our colleagues' perspective, that just kind of opens up this discussion quite a bit. But Claire, do you have anything else that you'd like to add to this as we sort of wrap up?
Yes. And again, I'm just going to reiterate the one thing that's extremely important, but not focused on enough, is hydrolysis and the importance of beta-glucuronidase. Common pain points are false negatives, screening results not matching the confirmation results, very high concentrations of glucuronides in pain care samples. You want to have a good enzyme that will hydrolyze all of the glucuronides in your sample. So then again, as Scott has mentioned, you need to stress test your analytical methods and your validations to make sure that your results are accurate. You need to test this with real patient urine, real pain care samples. So they have high amounts of glucuronides and high amounts of different drugs and flavonoids and everything else that's in urine.
One little plug for you guys. I'm not sure how much it matters now, but we all went through COVID. We all went through having these labs having to operate in the supply chain nightmare that we had to deal with and my staff had to deal with. We never had an issue with you guys. You guys had our back, had our customers back all through that. That says a lot. I mean, it's not only you can have a high quality product, you can validate a high quality product, you can have faith in a high quality product. If you can't get the product, it doesn't help you. So yeah, thanks for that. That was a huge benefit for us.
Awesome. Well, we really enjoy working with you guys. I mean, you guys are growing hand over fist. So it's always fantastic to see businesses be successful. And that success model comes with, that compassion, patient-centric model and high quality service. And I think, Scott, you encompass a lot of those features that you've highlighted in our products. Your services are fantastic.
Well, thank you.
I think your business philosophy, Scott, is outstanding. And that's the way I work in all of my businesses, especially when I was in the lab. I worked to make sure that we stressed and validated all of our methods so we had accurate results. And also in selling enzyme, I will not say anything that's not true. It's a matter of integrity because it's all about patients.
Well, let's wrap it up, folks. I hope you enjoyed this episode. If you want to stay connected, follow us on LinkedIn. And for more episodes, find us on Spotify or visit our website, www.imcstips.com. Catch you on the next one. Take care.